So I'm going to show you an overview of the Seurat pipeline. For those who are not familiar, it basically looks something like this. Give me a minute. Yep. Share. Okay. So this is basically what it looks like. So I see that a couple of you have used Seurat before. So we do have a mix of competency using this pipeline. Those who have not, don't worry, because this is really a basic workflow of Seurat, and I'm going to go through all the basic stuff. And for those who have a little bit of knowledge and competency, I'm going to add on a couple more advanced stuff for you to benefit from this class. And then learn this pipeline in a more thorough manner. So we start with our samples. Usually this comes in a form of count matrix. So previously you see that we generated count matrices for our experiment. So if you're able to combine the count matrices into one table, this can be a form of input into Seurat. If your data comes from a 10x genomics experiment, it will also generate several files, which we'll see today, which serves as input for the pipeline. Sometimes you may have a couple of samples, sometimes you have control samples, treated samples, samples coming from different time points, developmental stages, which we will look at today. You can actually import them separately and integrate or merge them into a Seurat object before processing it into the pipeline. So once this is done, once you have the object created, generally you will go through some quality control. You will see and filter out cells which are potentially background or potentially date cells, if you want to put it that way, and then perform several functions such as PCA clustering, or PCA dimensional reduction. We can then cluster the cells into groups and then do some sort of iteration to select clusters that you want to look at, and then perform 3 major downstream analyses, which includes marker identification to find which genes define all of these clusters. We can look at the expression of certain genes within each of these clusters and compare the expression of genes between 2 or more clusters. So we'll go through the entire pipeline that you see here, and I think it'll be quite cool. So we will be using a data set which was published, I think it was 2019, I believe. So the link, I will paste the link of the data set into Etherpad, so you can actually have a look at the paper. So what they did is to extract the cells from the cerebellum from different developmental stages. I think they started from E13 all the way to P6, if I'm not mistaken. And to perform single-cell RNA-seq on the different time points and present the data online. So what we did is to download a certain section of that data set, process it, and I presented you as a file that you've downloaded onto your computer. If you're not sure where to find such data set, you can have a look at their data availability section. They will usually spell out the geo accession number, the sort of accession that we have seen before previously, or give you some sort of link to download that particular data set through their FTP server or where it's found. We are going to use a pipeline which is very, very famous. I'm pretty sure anybody who has worked on single-cell RNA-seq begin with this pipeline. This is very comprehensive, very easy to follow. Like what I said, I'm going to inject a little bit more advanced stuff, a bit more personal touch. I think it requires a little bit more explanation on certain parts. So I'm going to do that, hopefully, clearly and something that you can benefit from. And there are several commands that you can actually look at when you are using Seurat. These functions are actually cool because it summarizes all the functions and processes that you can actually work with the Seurat object itself. And I will touch on a couple of those. this is a good reference if you starting to perform the pipeline on your own. Okay, so I think we can begin. Once you're ready, open up RStudio, and we will continue from there. I'm going to share my RStudio screen. To be here. Yep. Okay, cool. Give a thumbs up if you can see the my screen and whether the font size is big enough. If it's too small, let me know and I will increase it a little bit. Okay, should be fine. All right, so let's start by opening up an R script for those who are who didn't follow our first few workshops or not familiar with RStudio. You can open up a new R script using this button here, new file, and select R script. So we're going to type out the code that we will be performing today in an R script and run it into the console. So this is where the R script is, and this is where the console is, and the code will run in the console. We will start by loading. Give me a minute. Wireless keyboard always sleep on you if you don't entertain it for like 5 minutes. Come on, wiki. Okay, cool. So we're going to start with loading our dependencies. So this hash key or this comment is a character that tells our script that this is a comment line and don't perform, not to execute this line in our console. So you can actually comment out as much as you want within this R script and you will not perform any execution. So we're going to load 2 libraries. We're going to start by typing library followed by Seurat. All right, we're going to load Seurat first. And again, to run this line, you can click on Control Enter for Windows, or for Mac, your Command Enter. That should be fine. And you should see a message that says you're attaching Seurat object. And then this tells you that Seurat is successfully loaded. Another one that we're going to load is tidyverse. And I made a mistake here, library and tidyverse. Tidyverse is all small caps. All right. So this is the message that you should receive. Don't worry about these conflicts. There are, bound to be conflicts in function names in any. Yes.
So what is this? How do you get the hash key in it on a Mac keyboard?
Well, if you're using the UK keyboard, it's just beside your Enter key. So it's on the left of your Enter key. I think it's L. After L, you have this semicolon, and then you have the @ and then you have the hash on the right side. Do you see it?
Oh no, my keyboard is quite different.
Give me a minute. Let me see. Yours will be.
Not that one, is it?
No, not that one. No.
3.
And
3.
No, 3 is for the US.
Okay.
I think that there is a for UK using a Mac, so I can't find it as well. Actually, weird.
Yeah, so I'm
I can't find it.
yeah, it's not rated. This one, okay.
Oh yeah, there's another, but it's not shift. I think you try function. Try function.
Come on.
Function. Try function.
Function.
No.
There's a tree here.
Yeah, there's a tree, but you have to use a. You have to use.
In your face.
Oh,
yeah,
okay,
there you got it.
It's option, and 3.
Option. Okay, something new. So for Mac users whose hash is a shortcut, is actually option 3. So this is another shortcut for that. Interesting. I didn't know that actually. Thanks. Cool. Anybody has the same problem as well? Okay, it should be fine. All right, so we loaded Seurat and tidyverse, then we can proceed on with importing our single-cell RNA-seq dataset. So if you see my screen, this panel here, there are several tabs. This is my configuration. You might have a different configuration of tabs. If you click on files, you will see the folders and files in the current working directory. I'm currently now in home, where I have several folders like desktop documents. You should see a couple of these familiar folders as well. And in here, I also have the dataset that I have downloaded. Make sure that you unzip it. That means it's not in the zip format, and you are able to click on the folder. If you have not decompressed it, just decompress it now. No worries, it's going to be very fast. And in this folder, you will see 3 different subfolders representing the single-cell RNA-seq count matrix for the 3 developmental stages. So if you look at the E14, in here, you will find 3 files. The reason why there are 3 files is that this experiment is from a 10x genomics single-cell RNA-seq pipeline, and it outputs 3 different files. The key file is this matrix.mtx. This is the count matrix that we will import, but at the same time, it has information on the genes that are detected from the experiment, as well as the name or the ID of the cells in this file called barcodes.tsv. So with Seurat, you are able to load just the count matrix. If you just have the count matrix, you can just import that as it is. If you have a 10x genomics, there is a special function that you can actually use to import the entire contents of this file. So we're going to reference this file in our function. All right. So I'm going to import all of these files into RStudio into a variable. We're going to name this variable E14.crblm. This is just my name. You can follow it, or if you don't, if you can use your own name, it's fine. Just make sure that you use this name as we go along. So this is the name of the variable that we're going to create. Function is read 10x. So you can actually see that when you type read 10, you will see several options. You're going to choose read 10x, and the arguments, the main argument that you want to specify is the directory to the data. And because this E14 folder is part of the subfolder of GSE 118068, we're going to reference the path to the GSE folder first, which is in home. So you start with home, followed by GSE. You can autocomplete this again. If you remember, you can press tab. If you press tab, it will give you some options of folders, which starts with this naming. You can autocomplete by pressing tab again and select E14 as such. I've pasted this as well in Etherpad. If you can't or if you can't find your file, you can copy it from Etherpad if your folder is in home. All right. If your folder is not in home, you might have to find that folder in a different way. I'll show you another way in a while, but let's run this code first and see what happens. All right. So I'm going to press Control Enter for mine, Command Enter if you're on a Mac, and you can see that the command is running in the background and it's trying to read the 3 files and create a new object in R. So this is successful. All right. I'm going to show you another way if you can't find your file. I'm not going to run this, but you can try it out if you have problems. It's the same function, just instead of giving the path to your E14 folder, type file.select. File.choose, not file.select, file.choose. This will open up this interactive, read 10x. Right. It will open up this interactive user interface that you can actually select your folders. If you're able to locate your E14 folder using this method, do so. And then you should get the same 3 files imported into RStudio. It should be okay, I guess. Open. Yeah. I'm not going to run this, so because I don't want to conflict with what I've ran previously. I'm gonna comment this as well. Oh, sorry. All right, give me a thumbs up if you have e14.crblm successfully imported into RStudio. Good. Yep, we have a couple more who have not. Karen, you okay? Rewang, are you okay?
Sorry, I just have this file to be at home because I put it on the desk. So how can I just move
if it's in desktop, you can still use. Yeah, it's okay. Are you using file.choose?
No, I'm okay. I will try to choose
it's okay. I mean, if you want to make it simple for if it's in desktop, so you just have to change grid 10x from instead of just specifying this, you have to specify desktop first. So after home, specify desktop, right? And then followed by GSE 11.
Okay.
And this whole thing, right? Yep.
Thank you.
Yep. I will wait. I want to make sure that everybody gets this data set imported before I proceed. So I'm gonna attend to Fazal in a bit. Give me just 1 minute. Currently, this variable here, this object here is it contains cow matrix plus information, but it's not yet the object that we want to actually create. We want to create what we call as a Seurat object, and a Seurat object basically is an object in R that is very famous concept of object-oriented programming that you can actually create that consists of not only the matrices and information, but also all the calculations and the information that this particular experiment holds. So we're going to create this object first, and then I'm going to show you exactly how to play around, how to interact with Seurat object so that you can actually maximize the information that you can actually get out of this object. It's very interesting. So I'm going to create a new variable. I'm going to call it e14.seurat.crblm, right? Just to distinct from the other one, and create the function is called create Seurat object. See, it comes up as an option. Press tab to autocomplete, and I'm going to use input two arguments. One is counts, and you provide the input to the previous variable that we created, e14.crblm, without any open with the commas. This is an object, so we're going to provide this as an input. And then we're going to name this project E14. And there's a reason for this, which I'm going to explain in a while. All right. Again, the code has already been pasted on Etherpad. So if you're having problems typing this out, you can go ahead and copy from Etherpad directly. And let's run this function. It shouldn't take too long, because it's all has already been imported and compressed. So this function should be really, really fast. Okay. We're going to look at part 2 right now. We're going to inspect a Seurat object. Okay. You can easily have a summary of your project by typing out the name of your variable. So if you type out, e14.seurat, again, you can tap this to autocomplete the name. So it's very fast. So if you type this and just run this code, you actually see a description in console saying that this is an object of class Seurat. There are 27,000, close to 28,000 features, which are genes, 28,000 genes across 6,000 samples for this particular assay. We're gonna talk about what this assay means in a bit. So this is a good summary. If you have a combination of other data sets into one Seurat object, you will see an addition number of cells and features if there are more features detected. You can have a look at the cell IDs for your experiment. These can be accessed. Before that, maybe I should give you like a mental overview of this Seurat object before we move on. So this is, it's not complex. This object is not complex. It's very simple, just that it follows this rule of object-oriented programming, whereby you, instead of beginning with a function or a process, you begin with an object. So if you think about it as a crate, as a box, you are presented with a box which has small holes in there for you to put in all your information, and these holes are already pre-created for you. Just that you have to fill in the information as you go along, if you understand what I mean. And right now, it contains the count matrix, the cell names, the gene names. Right now, as we go along, we're going to add more information into this object, and we're going to do lots of processing on this object. Okay, so one thing that it contains is cell ID. So this, in order for you to kind of have a feel of what's the name of the cells, the ID of the cells that you have in this experiment, you can actually call out the names of the columns, because the column names of this matrix actually contains the name of your cells, right? If you type call names e14.seurat.crblm, and I'm going to head this. So basically, I'm going to show the first 6 cells, and the way I'm going to do this is by using this pipe followed by head. So if you're not familiar, this is a function from tidyverse or from dplyr that allows you to sort of transfer the information from the upstream function, so call names. It will be transferred into the next function, which is head. So you're going to essentially see the first 6 cell IDs. So if you run this into console, you're going to see the names of your cells. And you might think that this is weird. Why are the names in terms of nucleotide bases and all? And the reason is because this is exactly how we identify the cells. The cells are barcoded when you perform single-cell RNA-seq. Each of these gems, each of these droplets comes from one single barcode, and you identify each barcode as a single cell, right? You typically won't change this because these are unique barcodes for your experiment, so you just keep it as such. I'm gonna tap a little bit more so you can see my screen. So that's how you basically look at your names of your cells. You can also look at the names of your genes by typing, instead of call names, you type row names, because again, the columns are cells and the rows are your features, your genes. So if you type the same variable, sorry, Seurat, CRBLM, and as well head this as well, and run this in console, you see that the first 4 cells are named as such. One tip that I can give here for those who are preparing your own experiment, your own data set, is make sure that you have gene names as as features and not gene IDs. The difference is that you can have genes coming from different gene IDs because they are found from a different locus, but they are expressing the same protein, same gene, so they will be named the same as the same name. And the reason why I prefer gene names is because downstream we're going to identify mitochondrial genes. We're going to identify genes which are very specific to certain functions. So having gene names will give you the ability to identify these genes quickly, as compared to providing gene IDs, which is can be a bit cumbersome downstream. So I'm going to just write here that try to have gene names as feature names, for example. Okay. Okay so far? Give me a thumbs up if this is okay for you. Cool. Great. So earlier on I talked about assay. You can have in one Seurat object multiple assay, and an assay come from potentially different sort of packages that you use to process your single-cell RNA-seq. By default, if you were to type out default assay followed by, again, the Seurat object. Again, we are going to always deal with the Seurat object for today. If you type this, you're going to see that the default assay is RNA. It's the preset for Seurat. If you have another experiment and you're trying to process, not experiment, if you're trying to run another package and process the same Seurat object, you may have additional assays add on to it. And we're going to try this later on. I'm going to see what I mean when I say that there are a couple assays that you can actually switch. So now if you look at the object itself, you ask, so I have gene names, I have cell names. Okay, fine. So what are the gene counts look like in this object? How do I figure out where's the matrix in this object? Right? This might get a bit complicated, so the way you type this is not very direct, but you will figure out that this is very common with Seurat objects very quickly. So type this out with me, and I'm going to show you what I mean. So we start with the name of the object, followed by the assay. So type this square brackets twice. It's not once, it's double square brackets. And within it, type RNA with inverted commas, followed by @count. So if you type @, it will give you a couple of different slots for this Seurat object, and we want to look at the counts for now. And try to, let me see, should I head this? Let's try to head this because it might be a little bit big. Oh no, actually it shouldn't be. So try to head this and try and run in console, right? And the reason why I say it's not big is because it actually, instead of printing out the entire first 6 lines, which is actually a lot of information, you can imagine you have like 6,000 cells, it's going to print out technically all count matrix from all cells. But because Seurat keeps this count matrix as a sparse matrix, it's going to show you a more condensed information, whereby all these dots here represent the expression of the gene in the cell, which refers to 0. It's a null expression. There's no value to it. If there is a value, it will tell you the exact count for that particular gene in that cell. So this is a condensed form of your count matrix that you imported previously. You can also have a look at, let me just annotate this, view count matrix. You can also have a look at the metadata of your experiment. So a lot of time your experiment has information to it. It's not just blank information. For example, in this case, our cells come from E14 stage. We can actually add information to our cells. And later on, when we do clustering and we do cell identification, you can actually add on information to your metadata. So this is actually very cool. I use this a lot actually, and manipulate this as though it's a data frame. Right. So the way you view it is exactly the same how you view a data frame, just that the slot or where it's found in the object is here. Right. So this is how you type it without any character within these double square brackets, and you type head. Oops, sorry, I should be running it, not enter. Run this on console, and this is the current metadata that we have in our data set. The original identity, which is E14. This is exactly where we actually input the project previously. And 2 columns that specify the number of RNAs that we have and the number of genes that we have in each of the cells. So this is your metadata section. There is another way to do this. This is the same. I like this a little bit more because it doesn't deal with these square brackets, which I don't really like, because it's always confusing between square brackets. And if you understand R and our syntax, square brackets is usually subsetting, so it's a bit confusing. You can actually type @metadata. This is the slot for the metadata section, and if you type head, this will print out exactly the same data frame, just using a different access function. All right, it's the same code. Okay. Let me see, I'm going to skip a little bit here, and I will show you how to add new columns later on. Okay, no, let me do that now, so that you can actually practice this later on. So I'm going to add in a new column. So I'm going to add a fourth column that says region, and I'm going to use the character cerebellum as the value for this column. So treat this exactly as a data frame. So you start with E14 cerebellum. I'm going to use the same idea as in creating a data frame, which is to actually specify dollars and the name of the column that you want to create. So this is trying to say that I'm going to create a new column naming that is called region. And I'm going to assign the value of cerebellum. And you might be asking, why do I just provide one value here? Because when you provide one value, it's assumed that you're going to create a character vector of the same exact value for the entire list of cells, right? Run this, and you should get the fourth column with the value cerebellum. I'm just going to copy paste this, or you can run the same code again that you typed previously. So if you run this particular code, you see that now you have a fourth column called region, and it has cerebellum value for the entire list of cells. Okay. Okay, cool. Let's start with a first activity. All right, it's going to be simple. You can just reuse the code that we have used above. So what I want you to do, if you go to Etherpad, I'm going to paste the activity on Etherpad, right? And I want you to import the other two data sets. So we have E16 dataset and we have E18 dataset. So I want you to import into R and create Seurat objects for each of these dataset. You can name them e16.seurat.crblm, the same way as how we named it for E14. We can continue on from there. Well, I'll give you about 2 minutes. It's not going to be too difficult. If you see, that an object has been created and stuff, that means you're on the right track. Okay, about 2 minutes. Okay, so if you have tried it, hopefully it's you managed to successfully import the two data sets. I'm going to paste the code to the answer and to Etherpad. And if you have not imported, you can run this code. Again, if your file, if your folder is in desktop, do change the directory to desktop, followed by the extension and the subdirectories before proceeding. Okay, so I'm going to run this as well. Okay, I'm going to also copy and paste, save a little bit of time. So I'm going to run this whole thing. So if you can see, I highlight all 4 lines of code and type Control Enter. It will perform the entire set of code in Seurat in one go. Not in Seurat, in RStudio in one go. Okay, so the idea here is we want to combine the 3 time points, 3 developmental stages, into one Seurat object so that we can actually see how the gene actually changes in different time points, and this gives you some sort of meaningful developmental changes in expression. So we're going to start with combining the 3 Seurat objects. All right. So similarly, we will create a new variable. I'm going to make it a bit more simpler. Just name it Seurat.merge. Okay, this is the name of the variable. And the function to merge the 3 Seurat objects is called merge, right? Followed by this might be a bit tricky. So if you want, just copy this from Etherpad and run it. But I'm going to show you exactly what I'm trying to do. The thing is, you have to start with one object and merge additional objects into it. So I'm going to start with e14.seurat.cerebellum, and then the other argument is y, which is what you want to add on to it. And this is provided as a list if you're adding more than one object. So I'm going to specify e16.seurat.cerebellum as well as e18.seurat.cerebellum as such. This is optional. but you can also add cell IDs to it. Add.cell.IDs equals C. Sorry, I'm going to press enter so you can see everything in one line rather than very long line. I'm going to name it E14, E16, and E18. This might not be necessary because you already have the original identity from each of the experiments, so it will be combined. But I'm just gonna specify this explicitly so that we don't miss any information. So if you have pasted it from Etherpad, run it, and okay, so because I used the wrong argument. It's add.cell.ids. I'm gonna run that again. Not all .cell.ids. There's a warning that says here that check duplicate names. Oh yes, the other reason why you add the cell IDs is also more importantly because some experiments you are reusing the barcode. So you have X number of barcodes in one preparation. If you replicate this with different developmental stages, your barcodes will be reused. So by adding the cell IDs, you are able to distinguish the cell IDs from these three different experiments, and you can actually see that it's different in a while. I'm going to show you, well, I can show you now. If you go to call names, which is again printing out the names of the cell, if you type call names followed by Seurat.merge, the merge Seurat object, and you head it, this will have an extension, or we have a prefix called E14 to the cells. If you repeat it and show the tail or the back end, the last 6 cells, so I'm going to replace head with tail. So again, head prints the first 6, tail prints the last 6. You can see that the last 6 IDs has E18 on them. Okay, so these argument add.cell.ids differentiate your cells based on your given prefixes. All right, I want you to play around with this merge Seurat object in a bit, but specifically I want you to look at Etherpad and try out these 2 activities. It's very simple. These are basically multiple choice questions, and I want you to see if you can play around with this merge Seurat object and figure out what is the total number of cells in this new object, as well as the other one is what is the highest number of genes detected in a cell. So what is the maximum, or what's the maximum genes, not maximum, what is the highest genes in a particular cell? All right, so you might need to know a little bit of how to figure out what is the maximum value of a vector, but it's not difficult. It's the same thing as head and tail, just that you have to use a different function. So again, 2 minutes, try this out. We can, I'll show you the answer after 2 minutes. So I see that people have input the right answer for the first question, which is good. So one way of determining number of cells is just to type out the name of the variable. So I'm going to type Seurat.merge into our script and run it in console. And from the summary of the description, you can see that the number of cells, number of samples here is 19,401, which is good. And the other question, I guess this is maybe a little bit complex. I guess you're not very familiar with the data frame syntax, but the person who managed to get this correct, it is correct. The second part is to determine max number of genes. You can reference out the data frame and print out what's the maximum value for that column. So if you remember, we have 4 columns. One of it is called N feature RNA. So I'm going to type out Seurat.merge $n feature RNA. This is the number of genes in each cell. And instead of head or tail, I'm going to type max. This will print out the maximum value of that particular column. And if you type it out, answer is 7,168, and the answer will be 8. All right. Don't worry if you don't know this. This is again a tip or tool I'm trying to give. And if you're not familiar with R syntax, this is one of the many tools that R has to allow you to figure out the information within the data frame. I'm going to give you one pro tip here, but you don't have to run this. Basically, if you know the maximum number of genes, can you then figure out which cell contain that maximum number of genes? So I'm going to paste this into Etherpad, so you can try this on your own to find this cell. All right, I'm not going to run it today. Cool. So previously we checked the names of the cells in this merge object, and we see that the different cells from different stages contain this prefix, so that's good. That's a unique ID for each cell. Let's look at the metadata for this object. So metadata again is stored at meta, this Seurat.merge followed by add metadata. This is one way. The other way is to use that double square bracket if you want to. You can see that now, oops, sorry, I should run this, not press enter. It still has the same 4 columns: origin, identity, encount RNA, and feature RNA, and region, because we created region previously for E14, right? We created this particular column. But if you tail this and see what is the last 6 lines of this metadata data frame, you see that for E18 and E16, region is labeled as an A, and this is because we didn't label E16, E18 with the same value, right? Makes sense. So we can actually go ahead and fill up this metadata for the entire list of cells, and we're going to create region metadata for all samples, and we're going to use the exact same function as previous, just that now we're going to create it in this new merge object, not the E14 object, and you're going to assign the value cerebellum to this entire column. Okay, run this and let's check tail value again to make sure that now E18 and E16 also has this value cerebellum, and there you go. Okay. So again, this gives you a way to modify the metadata, and again, you can add any information. I'm going to show you a couple of ways to add other information later on, or even remove information. Actually, sometimes you create too much columns, and there is a way to actually remove some columns and simplify your data frame a little bit. Cool. There is actually a default identity to each cell. So you can actually assign your cells to one particular identity based on the column that you have in your data frame, or if you provide a new matrix or a new vector with identities. So currently, if you type idents, IDENTS, this is, I think, short for identities, followed by the name of the Seurat object, it will tell you the current identity of the cells. So I'm going to type levels here, because I don't want to print out the whole vector. I'm going to type out the level, so what values are there. So levels only apply for vectors. That means your values are discrete, and they can be actually grouped into factors. So with this, you can see that there's 3 identities. So you have cells which are from E14, you have cells from E16, or you can have cells from E18. So this makes sense, right? I will change this in a bit in the next part, so that you can actually see how you can actually label the cells differently depending on what sort of plots you want to create. And it's actually very convenient to kind of like go back to these identities often, so that you can manipulate your your preset identity to begin with. Okay, so great. We will now move on to the next part. So this is now in part, I think I lost count, should be part 4. My script says it's part 4, and it's quality control of our data set. And this is very important because you want to have a feel of the overall quality of your cells. You don't want to analyze cells which are, typically non-viable and do not follow a correlative pattern with other cells. You want to remove outliers, put it that way, right? And the first thing that we're going to figure out is to see which cells are potentially non-viable. And the way we do that is to actually calculate the percentage of mitochondrial genes in the cell. And this is, I guess, a very common proxy for viability right now, because the percentage of mitochondrial genes does correlate with the status of the cell. If you have too high a percentage of mitochondrial genes, it could either mean that the cell is under stress, so the number of mitochondrial genes rise up, or the overall transcriptome is downregulated or they are degraded, and mitochondrial genes percentage are now slightly higher. So this is actually a good proxy for cell viability, and we want to start by checking for checking if our Seurat object contain mitochondrial genes. And again, this is very important because, as I mentioned previously, if your matrix, your features are gene IDs, you have to figure out which genes are mitochondrial genes, which IDs represent mitochondrial genes. If you have data from, let's say, 10x or like processed data, like in this case, we are looking at gene names as features, you can actually fish out genes which start with a particular pattern. And usually mitochondrial genes start with this value MT followed by dash. I'm going to show you what I mean. You can follow along. I'm going to use this function called str subset. This is short for string subset. Again, code will be in Etherpad, so that you can copy paste from there. So I'm going to look at the names of all the genes and subset those genes which potentially start with MT. Right? The way we write it is like that: row names Seurat.merge. Row names because this is the row names contains the gene names or feature names, and the pattern that we're trying to subset for is like that: MT dash. Right? The first character here, this arrow pointing up, means that you want to find a pattern that begins with M and T followed by dash. Right? This is what we call as regular expressions. This is kind of a cool thing if you're familiar with it. So if you run this into console, it will try to figure out whether you do have genes starting with MT, and we do have 13 genes, which are annotated, and these are our mitochondrial genes. So we can use this list of genes to calculate the mitochondrial gene contribution to the transcriptome. If your data set do not have this MT prefix, try other combination. For example, I'm just going to copy paste this, and you don't run this, okay? I'm just going to comment this out. If there isn't MT pattern, you can try different combination. You can try capital M and T. Sometimes, depending on the nomenclature, some databases use capital M. Humans sometimes use M and T, capital M and T. So try out different combinations to see if you are able to fish out mitochondrial genes before proceeding with the next part. All right, I hope that is clear why we're doing this. Okay, so now that we have figured out that we have mitochondrial genes in our matrix, let's now calculate the percent of mitochondrial genes. And we will be using what we'll be doing is to create a new column in our metadata, that will calculate, will specify the value of the mitochondrial genes in each of the cells. So I'm going to type this slightly differently. I'm going to use seurat.merge, which is the name of our variable, followed by, let me use $% empty. Okay, you can type it as such. It should be, it's actually fine with the .mt here. It will be recognized as a string or as a name of a new column. And the function to create this, Seurat has this cool function that says that is percentage feature set. And the input is seurat.merge, the object that we are trying to calculate from. And the pattern is the pattern that we have asked for previously, which is up arrow followed by empty dash. Again, this pattern changes depending on your prefix for your mitochondrial genes. So you can change this accordingly to calculate mitochondrial genes. Run this in console. Okay, and that's it. Let's view our metadata again. Seurat.merge add metadata. I'm going to use add metadata slot and do a head. So again, first 6 lines of our metadata data frame, and you can see the 5th column that says %.mt. It should be fine. Okay. Cool. Give me a thumbs up if you are following through. Give me and so that I know that you're okay with this part so far and can follow through. Cool. Okay. Cool. So we can actually see and plot out the distribution of our continuous variable as different sort of plot. We can plot anything actually at this point of time. I'm going to plot a violin plot and look at the distribution of our genes, our RNA counts, and %mt as well, % mitochondria in our data set. Right. Again, the code will be in the Etherpad. Right. I'm going to just type that to create violin plots. Yeah, it should be okay. Of continuous variable. Right. So we're going to create violin plot. Again, always start with the name of the object, seurat.merge, and this is followed by the features that you are trying to plot. And features here, in this particular QC part, we are trying to plot the 3 columns. All right, we're going to start with creating a vector, and inside we're going to specify the name of the 3 columns and count RNA and feature RNA, as well as %mt. Right? If the names of your columns are slightly different, you can modify it to the names of that you have given in your columns. So run this again, Command Enter, Control Enter, and you will see a plot in your plot section, or plot grid here at bottom right. For mine, it's bottom right. So this is basically showing the distribution of the 3 different variables in your experiment. One thing which is weird here is that the data points are grouped based on its identity, which is fine. This is fine because we have previously created an identity based on the experiment E14, E16, E18, and it will create a plot with this grouping. Sometimes you want to revert back to an entity whereby it's homogeneous for entire list of cells, and you just want to view the entire group as a whole, right? And for us, it's convenient because we actually have a column called region, which is consistent across the entire experiment. So what I'm going to do here is to rerun this again, rerun the violin plot, but before that, change the identity. So we're going to see how to change the identities of the cell. So again, idents followed by Seurat object. This will actually call out the identities for each cell, but instead of printing this out, I'm going to override the identity and use the column Seurat merge dollars region. All right. What it's going to do is to reset the identities from E14, E16, E18, and label everything as cerebellum. Like the whole thing will be just having one identity. Right? Then we can actually see a much more simpler, much more overall view of the cell distribution. So I'll run this in your console. I'm going to copy paste the previous volume plot that we created and show you that now it will look slightly different. Okay, so it looks like this. So basically what we did was to kind of like simplify the plot a little bit so that we see just one volume plot for the entire experiment. So it's one way to change the default identity. So it's quite quite convenient. Okay. Cool. So you can actually use this for QC and have a look at the distribution of your RNA counts, your number of genes, percentage MT. You can see that you do have some outliers of cells which has very high MT, mitochondrial gene contribution. Typically, you want to keep to a minimum, or typically you want to keep to less than 5%, but that also depends on your data set. Some cells might have slightly higher mitochondrial genes, so you have to look at your distribution. And generally, the cutoff here is about 5, so that would be a good cutoff for a cell which is viable and has proportionally accurate number of mitochondrial genes. We are going to actually visualize this in a different way, right? I'm going to very quickly. This is let me see. I'm going to plot this feature scatter. So basically, instead of looking at a one-dimensional plot with each of these feature as on the y-axis, I'm going to put a two-dimensional plot, a two-way plot. I'm going to plot N-count RNA and %MT together with on one plot, and the other plot N-count RNA with N-feature RNA. So I'm going to paste this in Etherpad, and I think you will understand what I'm trying to say as we run the code. Right? I'm also going to paste this to save a bit of time. So if you run the code with me, it's already in there. Right? Run the entire thing. So again, you can run everything as a block. If you highlight these 3 lines and type Control Enter or Command Enter, right, it will plot out the 2 plots that you have here. Right? I think I'm missing this. I'm going to change the code a little bit so that you see this. So plot out this this code here. So basically, I created a feature scatter with and create a new variables to it, plot 1 and plot 2, and print out plot 1 and plot 2 at the same time into the plot. So it currently looks a bit squished. So if I open up the window a little bit, take a while. Yeah. So this is very important here because it basically tells you this two-way plot of the 2 different variables. It also tells you the correlation coefficient for each of this plot. For the plot on the left, this is %MT versus N-count RNA. You typically want a not too negative, but slightly negative value, which basically means that cells which has very low RNA count represent non-viable cells, and they should have higher %MT metric. So it should be high on the top left-hand quadrant, and it should be low in the bottom right quadrant. So the slope will be slightly negative. So currently the data is actually good as it is, because it's a negative value, and you see a good correlation of number of RNAs and %MT. So this is fine. On the right, it plots number of genes on the y-axis against number of RNA, and this should be positive correlation. So cells with high RNA count typically have higher number of genes, because that there is some sort of correlation with the number of reads that you actually detect with the number of genes from that cell. However, you see that the correlation coefficient is it's good. 0.9 is actually very good, but we can actually bring this on a little bit higher if we remove this outlier here at the bottom left. So this is cluster of cells which has very low number of RNA count as well as very low number of genes. So if we remove this, we can get a better correlation coefficient, and that's exactly what we want to do here, all right, in a bit. So we're going to subset the data a little bit. We're going to filter it. But before that, I want to show you that you can actually treat this plot as a ggplot. If you're familiar with ggplots, you can actually add on additional functions and commands so that you are able to plot out, let's say, lines or text, or even modify the colors as it is. So this is the cool thing about Seurat. It doesn't create its own set of plots. It uses ggplot, so you can modify them as you go along, actually. So I'm gonna quickly, just plot out a line in plot 2. I'm gonna copy this set of code, but follow along with me and just run the second code. Okay, I'm going to paste it into my RStudio. So just print out a second code, plot 2 plus geom h line. So basically, I'm just trying to say that run this. Okay, and if you run this into RStudio, you can actually see that you can actually draw 2 additional lines, one which is a vertical line, the other one is a horizontal line at this intercept, 500 and about 21,000 on the x-axis. So you can actually see, and modify this value a little bit to a point that you are satisfied with your population of cells. If you can actually change these values so that you can create a better cutoff if you wish. But I think this is a good cutoff, 500, because it separates this very weird cluster from the rest. And also, I'm also trying to not take these cells with too high number of RNAs, which do not increase the complexity of the transcriptome, because it's sort of plateau. And I think this is an outlier, but this is personal. This is my personal opinion. I prefer to see a more clustered set of cells and look at them. So what we're going to do here is a couple of things. We are going to filter our data set a little bit, and we're going to select cells which has a minimum genes of 500, so anything above the y-axis of 500 and anything lesser than 2100. And also we want to select cells with percentage MT of less than 5. So as mentioned, we will use 5 because the whole distribution centers around 2.5. So below 5 seems to be a very good value here. So function here is subset, and we're going to overwrite our merge data. So take note that once you perform this, and if you overwrite your variable, you cannot retrieve the cells that you have removed unless you run again the pipeline from the top. So I'm fine with this. If you're not fine with this, you can create a new variable to save this filtered set of cells. I'm going to override this just for simplicity for today's workshop. Function is subset. And again, the code is a little bit complex, so you can go ahead and paste it from Etherpad. You start off with the name of the object, Seurat.merge. Subset here is less intuitive because it's a mixture of a lot of things. But basically, you want to specify which column you want to subset from, as well as the value that you want to set as a threshold. So first is N feature RNA. Number of genes in the cell has to be more than 500. I'm gonna, and I use followed by, sorry, it's not comma, it's actually N, because we are actually doing multiple filtering parameters. So it's N. I'm going to press enter so that it sort of goes down and you can actually see it a little bit more legibly, followed by N count RNA of less than 21,000. So again, this is the cutoff for the whole x-axis, less than 21,000. And last one, which is percent, sorry, not comma, and percent MP less than 5. So 3 parameters to filter for, and this will override the original Seurat object. Okay, run this. It shouldn't be too long, although we have quite a number of cells, but it should be quite fast. And now if we were to check the plot again, so I'm going to copy paste the entire plotting, the feature scatter, the entire 3 lines, take the whole 3 lines and run it again, because we reuse the variable name, so we can replot this again. You can actually appreciate that now the correlation coefficient for the graph on the right, the plot on the right, is better. It's quite close to 1. And on the left, you still have this negative correlation of percentage mitochondria and N count RNA. And I think this is a good subset of cells to use. Okay. Thumbs up if this is okay, or if this is too fast, if you need me to repeat certain parts. We're going to go for a break in a while and just run one more function, and then once you run, it's going to take a while, so we can take a break. So we're almost on time. I hope it was like 10 minutes earlier, so we can have like slightly more comfortable break. We're still going to have 15 minutes break, just that it's not at the time that I want, but it's okay. So we have the cells that we want. This is, I would say, high quality cells. I would consider this as high quality, and we're going to next normalize the data, and part 5, normalize, scale, and identify high variable features, genes. Okay. A conventional pipeline, if you look at the link that I gave earlier on, it breaks down these 3 different parts in 3 different sections. So you start by normalizing, identify high variable genes, and then you scale. All right, which is fine if you understand what you are, or the method of normalizing, the method of finding these variable genes. I've recently moved on to using one function, which is actually like a wrapper function that the people from the Satya lab actually use, or they actually create this function, but it's from a different package. Don't worry, you will have this package because it will be installed part of Seurat. Just that it's actually not Seurat, it's actually a different package that they create, but it will be loaded together, if you understand what I mean. And the function is scTransform, and this function scTransform will do these 3 processes in one go. It will take a while because it will run, so we can have our break while waiting for the function to finish. But essentially, you do not have to think about the exact variables, because it will look at your data set and pick out the best method to normalize, to scale, and method to identify variable genes. This is a bit more automated and depends on your data set. If you want a bit more control of your variable genes and your scaling, do refer to the link that I gave earlier on to perform these 3 parts separately. Okay, so again, we're going to override the variable name, and function is scTransform. It's very simple, scTransform, followed by the name of the Seurat object, and I'm just gonna do one argument, which is verbose equals to true. This will not change anything, just that you will print out the processes, the normalization, scaling, and finding variable genes as you go along, so that I know what sort of output it provides and whether it's successfully executed or not. Okay, so run this, and we should be, if you see, this message that says calculating attributes and stuff, means you're on the right track, and we can have our 15 minutes break. while we wait for this to finish, and we can proceed on after 15 minutes. So time now on my watch is 11:32. Break starts at 11:30. I put 33. Resume at 11:48. All right. If you have any questions in the meantime, I'll still be here, and we can discuss and we can rectify some problems that you have. If not, I will see you in 15 minutes, and I'm also going to get myself some hot drinks as well. So I'll see you in a bit.